sorry for the delay. I'm really bad with technology. So, so I'm Kathryn Chepiga, and I actually did my senior thesis on my summer internship research that I did. I worked at Johns Hopkins this summer, and I worked under Dr. Paul Watkins in his lipid enzymology lab. And what that is he studies enzymes that break down lipids, which are fats. So fats are used in a variety of different biological processes, and as you can see here are listed all of the different biological processes that fatty acids are used in. But the interesting thing is that fatty acids can't directly be used without being first converted into fatty acyl-CoAs. And I'm not going to go into all these processes or what fatty acyl-CoAs are, but as I said, we studied the enzymes that manipulated this process. So the family of enzymes that we focused on were Acyl-CoA synthetases, which I will abbreviate as ACS, and they are needed to first convert fatty acids into acyl-CoAs and to do all of these metabolic processes that we use every day. So I was really lucky in that in studying these enzymes, they found a really interesting finding. They were just trying to see what cells these enzymes are in and what kinds of levels they were in. And one particular enzyme of this family, called ACSVL3, which stands for very long chain acyl-CoA synthetase 3, was found in really elevated levels in different types of brain tumor cells. So from this slide you can see the first row is normal brain cells, and we did a staining for the different enzymes. So in the brown you can see the staining for the enzyme. The first across is ACSVL3, and then ACSBG1, which is also in the same family of acyl-CoA synthetases, is shown below. And you can see it's pretty much consistent with the ACSBG1 from all different kinds of cell types, that it's at a pretty consistent level, but from normal brain to malignant different types of tumors, the ACSVL3 was elevated in all these different types of tumors. And the type of tumor that we decided to focus on were glioblastoma tumors, and this is because they are the most malignant and most aggressive kind of brain tumor. Also, we were working in the Kennedy Krieger Institute, which is primarily focusing on child diseases, and glioblastoma tumors unfortunately affect many children. So, once again, this acyl-CoA synthetase, ACSVL3, was found in very elevated levels in this type of tumor. So our hypothesis was that hopefully in studying this enzyme and maybe better understanding its function, we could test, first of all, if it were inhibited, if it would hurt normal cells, or if it could specifically only kill the malignant cells. So we wanted to find a drug that could specifically inhibit this one enzyme without killing normal cells, but only killing the malignant cells. So, what we did was we did an acyl-CoA synthetase assay, which we look at the cells and then we determine the level of acyl-CoA synthetases in the cells. And so in the yellow you can see this is the control, and the yellow is the amount of the total acyl-CoA synthetases. And we wanted to bring that amount down to the level of what the ACSVL3. So the knockdown cells show, so you see the difference between the yellow and the orange in the first row is all made up by ACSVL3. So it shows that ACSVL3 is a very large percent of all of the acyl-CoA synthetases in that cell. So we want to bring the yellow bar down to the level of the orange bar. And these are all the different drugs we tested. We chose a really small drug bank based on drugs that were known to inhibit a structurally related enzyme, which is ACSVL2. And we chose 20 of those drugs just because of time, because I was only there for a couple months this summer. but in the future they'll be testing the rest of those drugs in that same drug bank. And all of these drugs, I don't know if you've ever seen the names before, but they're used for, they're currently being used for psychological disorders such as schizophrenia. So they're already on the market, and they're known to be safe and hopefully get across the blood-brain barrier because they're being used for a similar disease. So the one that we found to be most effective was the one in the last column, the triflupromazine, because it was specifically able to inhibit ACSVL3 alone, as you can see by the fact that the orange bar remained at the same height. But the yellow bar came down a bit, meaning it specifically inhibits some of the ACSVL3 present in these cells, but it unfortunately wasn't able to inhibit all of it, and we wanted to see what would happen if we had a drug that could inhibit all of the ACSVL3 present in these cells. So future research is going to be done using this drug, seeing maybe at different concentrations if the concentration were increased, if the patient could tolerate it. But that's what my research was on. And I'd like to thank Dr. D for doing a lot of research with her, and Dr. Watkins and the lab at Johns Hopkins. Any questions? Yes?
Did you happen to partake in the current treatments for glioblastomas now? Looking at how they treat the actual tumor now?
No, we didn't do any of that. Actually, well, since I said it was a lipid enzyme lab, they usually don't even do cancer research at all. They do this really esoteric work that a lot of people don't really know about. They just like to understand the function of different lipids and see if they're involved in anything that is relevant to diseases. But they usually don't even do cancer research whatsoever. So this is all really new. This was within the past 2 years, so they didn't explore any current treatments. And also they were looking for drugs that would specifically fit the enzyme that we want. And so I don't think that a drug that we're currently on the market would work on this exact enzyme, but that would definitely be interesting.
And did they speak on the prognosis in your studies of folks with glioblastoma?
You mean if this would work, if this drug would work on them?
The current prognosis, did you speak on that? Was that part of your study as to once diagnosed, what's the prognosis of those patients?
Oh, no, I don't know. And all the testing we did with these drugs was it just in test tubes, so in vitro, and no in vivo testing has been done yet with these. Yes?
Maybe could you let all of us know exactly what your plans are for the future, and if you want to continue research like this, or what ideas you have?
Yes, well, so this research helped me, put me in the direction of what I'm going to be doing next year. This is more biochemistry, and I also have done a lot of organic chemistry research with Dr. D here. And so next year I wanted to do a combination. I really liked searching through the drugs and testing different drugs as inhibitors for diseases. And I also like the organic chemistry research. So I'm going to be doing an organic chemistry PhD program at Emory in Atlanta next year. And I'm hopefully going to be placed in a medicinal chemistry lab. So get a little combination of both.